For the last, but not least, presentation of today, so we're gonna have Stephanie Ribet. Here she introduced herself in the beginning. So, but before she showed some like nice features and data processing using Python with 4D for 4D STEM data. I want to give a brief introduction about like different strategies for 4D STEM data processing. So as I mentioned in the beginning, so as the usage of 4D STEM scanning diffraction for like different groups around the world, so there are many packages available these days. So let me, if I can move my slides. There we go. So some of them are highlighted here. They all are based, I believe all of them are based on Python. Yeah, they're all based on Python here. So you go for HyperSpy, that is known for like the spectroscopy analysis, spectroscopy here, like PyStem, PyXam, LibreTem, and most recently like Py4D STEM. So they all like open sources, and they all have nice functions to apply like virtual detectors, from virtual detectors up to like string mapping, and you can do also use those packages in conjunction with digital micrograph, right? As Anna showed earlier, you could use that Python interface from DM to run those packages. But one thing here that I want to highlight, especially for Stephanie's talk here, is that if you want to do write your own code to start analyzing those data, you need to dig deeper here, right? So you could go straight, go ahead and use those packages that are ready to go. You might have to modify them, and they are open sources. You can do so. But if you are afraid to start, if you think that's too hard, so the best way to start is from scratch, right? So that's our plan for today. And then Stephanie will use, and she'll talk more in details when she shares the Jupyter notebook here. She'll use most of like SciPy libraries like NumPy and MATLAB here for plotting. So that's where you want to start, right? So, and then she'll share these strategies with you all to for your first step on analyzing this data. So again, so feel free to ask questions. I'm gonna be checking the chat, and if you have any other comments, also you can address later. So before we start that, as I mentioned earlier, so there are few strategies for data management, right? So something that is important before you even start your processing. So three of them that I want to highlight here is, and then we talked a lot about them over the last few hours. Is one is like binning the data, right? So you may not need a lot of pixels. You might reduce your data size just by binning, right? The second one here is cropping the data, right? You might not need like a super large area like I show on the top part here. You might just want to crop a few pixels, and that you gain in terms of size. You may not gain in one image, but the moment that you acquire thousands and thousands of diffraction patterns, that will definitely help. And the third one is counting, right? So the K3IS does count already electrons, so you could convert your diffraction data in a vector with like ones and zeros, where one is just the index of your like pixel size in the diffraction pattern, showing that an electron actually hit that portion of the detector, and that helps a lot by like reducing the data size, and that might help you to not only mine, but it will definitely help you like in terms of transferring data and carrying the data and analyzing in the future, and especially storaging too. All right, with that, I'm gonna give the desktop to Stephanie so you can share.
That is good. Thank you, Roberto. So the first thing I'm going to do is drop in the chat a link. I'm going to share my notebook and my data for this session with you. So feel free to download it now and follow along or do so later. And then I will share my screen. Here. So in the workshop today, I hope that my goal here is to convince you, as Roberto just explained, that with just a little bit of coding and using some really basic packages in Python, we can be able to analyze our four-dimensional data set and get some really interesting results. We'll start by doing some things that are very similar to what Amna just demonstrated in Digital Micrograph, and being able to tune our analysis to the diffraction pattern. And then I hope that I can convince you that doing it in Python also allows for a lot of creativity and flexibility. So we'll show a little bit of some more advanced analysis type techniques. We are going to be using basic packages like Roberto said, and then with a little bit of help from a scientific imaging processing software. And then also we're going to use some software from NSEM just to be able to load our data into Python. So basic outline of what I'm going to go through is first just how we load the data into Python, then some basic cleaning, kind of like Roberto just talked about, and then we'll go through the analysis in two stages. First, some more basic analysis, and then going to some more advanced techniques. I'll pause in between, Roberto, if you want to see if there are any questions or confusions that we clear up between steps, if there's anything that comes in the chat. So as I explained, the first thing we're going to do is just load our packages, which are here. And we are going to rely a lot on NumPy and Matplotlib. NumPy can be used for analyzing your array and turning our 4D data set into 2D images that we can understand. And then Matplotlib is the main plotting tool in Python. So we're going to be using that to look at, to make our maps. The next thing we're going to do is, and this is one of the first challenges with analyzing 4D data in Python, is how do you actually get your data into the notebook? So most of the time when you're doing your analysis, all you really want is your 4D array, and you want to remove everything else like the metadata that is stored in your file, and you'll add that at the very end for your scale bar and plotting and things like that. So there's a bunch of different packages and softwares that are microscopy specific. I'm using NSEMs here, but there's a lot of different options, and you want to extract just the 4D data structure, which is what we've done here in this kernel. I also printed out the shape of the data, which will be helpful for me for understanding the size I'm working with. As we talked about today, 4D data, right, has 4 dimensions, 2 in real space and 2 in diffraction space. So the first thing I do immediately when I load my data is I want to understand an overview of what each of those sets of dimensions looks like. So I can get an average image by averaging all of diffraction space. So in this case, diffraction space is average 2 and 3, and I'm using a basic NumPy averaging tool to be able to average my data along those axes to get a virtual image. And then I kind of do the opposite to get an average diffraction pattern. This is the diffraction pattern for all of your image, and we're averaging over all of our real space dimensions, so axes 0 and 1. Going down, now we can use Matplotlib to plot them, and I'm going to show you them here. So this is how we're calling Matplotlib, and we're plotting our average diffraction pattern and then our average real space image. Keep in mind this real space image is basically a linear combination of bright field and dark field, so it's not that helpful, or so the contrast maybe is not the most useful, but I like to do it first to get kind of a lay of the land and start to understand the shape of my sample and some features that might be of interest. So this is the end of the first section where we're just talking about loading the data and getting a basic overview of what's going on. And we've used some basic tools in NumPy and Matplotlib to be able to start taking a look at our data in a Jupyter notebook. And Roberto, I'll pause to see if there's anything you want to add.
Yeah, I have a question here. So you talk about like loading the data, right? So most of the extensions, I mean, especially the recent digital micrograph data set. So you save the data in like the DM4 extension, right? So that can be loaded not only in Python, can be loaded like say MATLAB or using other package like Stephanie mentioned. And but one question that I have is this: what is the initial format? If you can go back a little bit on the 4 dimension. So yeah, so what those like 213, 342, 128 represent?
Yeah, so these are going to be our dimensions. So basically when I call data.shape, I'm asking Jupyter to give me the shape of my data set. So my 0th axis is going to be 213 and my first is 342. And those are our 2 real space dimensions. So if we go down to our image here, we can see it's 213 here by 342. And then the next 2 numbers we got were 128 and 128, and that's axis 2 and 3. And that's the shape of our diffraction pattern, which is right here.
Okay. All right.
So I'll move on now to the next section of the notebook, which is about kind of cleaning your data and reducing your data size. So as Roberto has mentioned frequently in today's workshop, one big challenge with 4D data is the data gets really big really fast. And one advantage of working in a Jupyter notebook that I didn't mention previously is you can run it on your own computer without any special software, which is great. But if you start having a bigger file size, you lose that advantage. And of course, there are workarounds, but a lot of cases you don't actually need a data file that big because 4D data contains a lot of redundant information. And depending on the type of analysis you're doing, you can use a smaller data set. So in our case, we bin the data because we didn't need the full resolution for this analysis. And that was done before I loaded the data in the notebook. And Anna showed some tools to do that in Digital Micrograph. But the other thing that we can do is cropping. If we look at our seabed pattern up here, there's a lot of space that doesn't contain any information. And what we want to do is look at just the diffraction area. And to make our detectors in Python, we're going to be using for loops where we iterate over all of diffraction space. And if there's diffraction space that doesn't contain any information, we're wasting our computational time. So the first thing I'm going to do now to clean my data is actually just crop it. So I'm getting rid of the portions in the upper left-hand corner that contain no information. And then I'm going to replot and recalculate my average image and plot them down here. So as before, I'm using NumPy averaging tool and then Matplotlib to actually show what's going on. And these images are reproduced here. So depending on the type of analysis you want to do, and I saw some questions in the chat earlier, you might want to do some other sorts of cleaning or filtering of your data before you do reconstruction. So whether it's getting rid of erroneous signal with a filtering technique or normalizing by vacuum probe to correct for any sort of deflection or inconsistency in your beam, we could do that here, and this would be the place to do it, but it's not where we're going to do it today because we don't need to for this type of we don't need to for this data set. So I'll pause again, Roberto, before I start actually doing the reconstructions to see if there are any more questions that came in.
Yeah, there was a quick question about the metadata. So when you export the DM4, when you save the DM4 from Digital Micrograph, that usually comes with the metadata that you definitely can read from like Python, right? So with any of the readers that use like either HyperSpy or NSYMPy in this case. So you could see it. So you have to read instead of, if you maybe go a little bit up, instead of doing your EM0 data, there is another section of that you can read like just basically like the metadata of that. So yes, it is accessible. That was the question.
Yeah, exactly.
And
So
from
this
that,
is the line where I selected that I just wanted the data, but
yeah.
like you just said, Roberto, we could grab any part of the metadata.
And that will be useful for like plotting your like calibration, have the calibration and other information that you may need to plot, like the scale bar, etc.
Yeah, exactly. Okay, so I'll move on then, and now we're ready to actually start doing our reconstructions. And we have seen how we can do kind of bright field, dark field, ABF reconstructions in digital micrographs. So we're going to repeat and do the same thing now in Python. And then kind of also talk a little bit about how detectors can be tailored to a specific diffraction pattern to highlight certain phases. So the steps we're going to go through in this section is first to find the center of our diffraction pattern, because I want to have my masks or my detectors aligned to the center part of my diffraction pattern. And then we're going to create binary masks. So basically what this is going to do is filter diffraction space. So instead, since we don't have a physical detector, we can create binary masks that overlay diffraction space and filter certain areas, and then we're going to integrate. So hopefully this will be clear as we walk through a few examples. The simplest case is bright field, so we'll start with that. But the first thing I'm going to do, like I said, is find the center of the diffraction pattern. There are a ton of tools to be able to use this. I'm using a scikit. This is a scientific imaging processing library and a tool they have for finding the center of my disk. And I was able to select this disk over all the others, paying attention to the thresholding. But there's a lot of methods to find the center, and also you could set manually. And then you want to plot, of course, to make sure it matches up to where you're expecting. So now we're going to create our bright field image, and we're going to create this binary filtering mask like I just mentioned. So what we do is we create indices. These are XY indices that go over all of diffraction space. So it's, again, I'm using this shape tool to find the shape of my diffraction space. And then my mask I make by creating a circle and then using an equality less than. So I have it plotted down here. In the first of my subplots, I have my diffraction pattern. And then in the second one, I have my mask. And basically this is an array of zeros and ones, and only the information right here is I'm going to let pass. And then in Python, there's lots of cool plotting tools. So I make use a lot of this alpha. This is the transparency value of my map. So then I can actually overlay my mask on my seabed pattern to see much more clearly the information I'm collecting. And we can see that it lines up very nicely with our central disk. So now we're going to integrate it. As one way to think about an image really is just kind of an array filled with numbers. So what I'm going to do is I'm going to create my bright field image. First, I'm going to initialize it. So I'm going to create an empty 2D array and fill it with zeros. So that's what I'm doing here. I'm making a NumPy array of zeros of the size of my image I'd like to create. And then I'm going to use 2 for loops. This is why it was so good that we cropped before, to run through all of diffraction space. And wherever my mask is true, I'm going to add that into my image. So what I'm doing is basically integrating this masking area, and I end up with an image that looks like this. This is my bright field image. And as you can see, this is a lot nicer than the virtual image we collected before, and we can start to see now some of the interesting features in this material, whether it's dislocation, those are different things going on in the matrix. So to recap, what we did is we found the center of our diffraction pattern. We created a circle and integrated that area, and that gave us a bright field image. Let's do it again now, and we're going to do the same thing, but we're going to do dark field. And the only difference between dark field and bright field, and makes dark field slightly more complicated, right, is dark field is an annular detector instead of just a disk detector. So this is where we build our mask, and I'm going to create 2 circles, one greater than and one less than, and then use a logical statement. So basically saying both things need to be true. So that selects just the area in between those 2 rings, like I've shown down here. So I've basically created a detector now that is between these 2 rings and this is going to be my dark field detector. You could tune this. So this is one power of scanning diffraction, right? Same thing as in digital micrograph that Anna showed. You're setting your dark field to be just right outside your central disk, but you can change your inner and outer diameter. So from the same data set, you could do a HADF image or a mid-angle dark field, a low-angle dark field. Here I'm integrating all of dark field, most of dark field region to get an image. So as before, we create our mask. I plotted it to check it. I initialize my image, and then I integrate. And I end up now with a dark field image, and I can plot it next to my bright field. And I have these two images side by side that show me different information about my sample that I got from the same scanning diffraction data set in Python. So now we're going to do the same thing again, but we're going to do it slightly differently. We want to look to get a little bit more information about the sample, and we're going to use detectors. We're going to do a sort of special type of dark field where our detectors are tuned specifically to our sample. And again, this is much like what Anna showed to do in digital micrograph. So we're going to look for precipitates in the sample. So with a little bit of prior knowledge about the sample, we expect there to be some precipitates and for them to have reflections where there are not reflections showing up in the average seabed pattern. So I'm going to build a detector that looks much like our bright field detector, but I just shifted its position a little bit. So it's not on the central disk and it's not on one of the main reflections, and I'm going to integrate this area. So this is exactly the same as the bright field analysis, but I just shifted a little bit its location. Then I integrated it as before, and I can plot my two results side by side. So here I have my bright field image, which shows the features we discussed already, and then a second map, which shows my precipitates. So this is the second phase in my sample, and the bright regions are showing me where they are. Just to make this a little bit more robust, what I'm actually going to do is repeat this analysis 4 times. So I'm going to build 4 circular detectors, which go around the outside of your central disk, 1, 2, 3, 4, and integrate each of them. So this kernel looks a little bit longer, but it's really just the same thing that we've been doing repeated 4 times. So nothing new is added here. And then we can plot all 4 of them. So we have these 4 resulting images. They have very similar information, but maybe slightly different. And you could argue whether there's some sort of symmetry breaking in your sample or whether it's just that I didn't align the masks perfectly. So that's analysis for another time. But what I'm going to do for simplicity for today is just sum them. So I'm going to end up with something like this. I've now created a detector that is 4 circular disks that are not connected that go around my central disk. I sum them and I get this image to compare to my bright field with very strong contrast that shows where the second phase is. And I think this starts to show some of the value of writing your own code and doing it in Python. I've created a detector that's not physically realizable in the microscope to be able to get some really interesting, important information about my sample. And I've done it with some really basic code, creating these simple shapes and using them as a binary filtering mask to assess my sample. And just before we conclude this section of the code, I'm going to take a quick detour to talk about plotting. So one value in Python is that you can have a lot of control over how you're actually plotting your final image. And that's maybe interesting for aesthetic reasons, but also very helpful for starting to understand and analyze your sample. So what I'd like to do is actually overlay these 2 images on top of each other to get a better idea of how these features interact. And that's not possible with a traditional color map because as soon as you put one image on top of the other, you block out what's underneath. You can use this sort of transparency trick that I've been using so far, but as soon as you put one image on top of the other, you lose contrast for what's underneath. So what I'm going to do is actually create my own color map. There's a great library of color maps available through matplotlib. But what you can do is you can download the color map and modify it. So I'm downloading the color map, and a color map is really just a vector of colors, and each color is represented by 4 values, red, green, blue, and then transparency. So I modify the last value in that vector, the transparency value, and then I add my color map back. So now I can create a really nice and helpful image where I'm plotting one on top of the other, the precipitate and the matrix. So this is helpful both for analysis and for representing your data. So I've been through a bunch of things. So Roberto, why don't I pause again and see if there are any more questions or things you want to comment before we do the last stage?
Yeah, a few questions here on like data size and to run on your computer. So if you can share some of the specs that you're working now. So in terms of like RAM that you have or and the size of the data.
Yes. So the data size is about 1 gigabyte. This computer is a 2019 MacBook Pro, and I think it has 16 GB RAM. My longest kernel in this notebook is about 90 seconds.
Okay, yeah, that's good. And then I guess to add to that, we have prepared the data beforehand so you can run this faster. And then as you can see here on Stephanie's demonstration that you do get a lot of information even after beating the original data. So the original data, if I remember correctly, was around somewhere around like 8 GB. So 8.
Yeah, that makes sense because it was binned.
Yeah, it was binned by 8, right? Yeah, yeah. And then that was yeah, and it's 1 GB. There you can run. It runs pretty well in any like standard laptop. And another thing here, if you want to comment on, is that in terms of speed, you are basically what kind of mathematical functions you are applying here. That I think that's kind of answers the question, because in terms of speed and usage of memory, so you do depends on what like functions you're using here.
Yeah, exactly. So the different, yeah, I think that answered it. It depends on the function. So a lot of the averaging steps take more time, but the masking and running through to kind of integrate this is surprisingly quick.
And there was a comment here also I mentioned earlier about other packages, right? They like for example HyperSpy or like 4D stamp picroscopy. So they do have functions built in. So and an idea here, and then what Stephanie is demonstrating is that you can do yourself. You can just go in and modify those functions, and then have those functions modified to answer the questions of your sample, basically. Right?
Yeah, totally. So same thing. So yeah, so all these packages can do some very similar things to what we've done here, and different challenges. There are different challenges when it comes to writing your own code and the learning curve for that versus understanding someone else's code and the learning curve for that. So it depends what you want to do and what for your analysis.
All right, that's great. We continue.
Okay, we continue. So this is the last stage of the workshop. And this is where you can really start to see how creative you can be in Python. And as we've talked about a lot today, there's so much different types of information that's contained in a 4D data set and all kinds of reconstruction techniques that you can use to really start to assess it and understand what's going on. So we're going to do two simple types of analysis, one starting to look at in-plane rotation along the z-axis, and the other to do something sort of like DPC that we just did in digital micrograph to look at beam deflection. And so I will start with rotation, and this is a kind of a long kernel. So what I'm going to do is scroll to the bottom and show you where we're going and then go back through and explain how we got here. So what I want to do is build an annular or ring detector, much like the ones we had above, and it's going to be a ring just like this. And then what I'm going to do is unwrap it and create a line profile as I'm shown down here. And the reason I want to do that is because I want to find where the most intense pixel is around this ring, or where the maximum is. And you'll notice that this ring lies just on top of the nearest reflection outside the central disk, but you could change that if you were interested in studying a different one. So what I'm going to do is for each pixel in real space, I'm going to take its diffraction pattern and use an algorithm like this to find the most intense pixel, and then plot all of that in a map. And that'll give me some idea of what the relative rotation of my sample is, and how nearest pixels are rotated relative to one another. So even though this looks maybe a little bit more complicated than what we did above, it's pretty similar in terms of scope of coding and different types of computational time. This is the longest kernel. Like I said, it takes about 90 seconds to run on my computer. So the first thing I'm going to do is take one diffraction pattern and use it as my test. I set this flexible so I can change it. And my advice would be that as you're developing your own code and creating methods like this, to leave this flexible and to test your method on a lot of different pixels from different parts and different types of sample areas within your whole sample, because before you run on the full data set, you want to try and minimize error and erroneous information. What I'm going to do here is select my number of dots. So I picked 90. There's a trade-off, of course, between your resolution and maybe how many dots you have in the circle and computational time. And then I'm going to convert these dots to angles, or right here. And then I set my radius before, like we talked about, and we're using the same center of circle from before. We're going to initialize vectors to fill with information, and then use basic sine and cosine functions to calculate those x and y values. And maybe, Roberto, this is what you were asking about before about different functions. So these are built-in sine and cosine functions from NumPy, and they're also pretty quick functions for running. And then what I'm going to do is, after I found the x and y values for that circle, that magma colored circle from below, I'm going to convert them to integers. And the reason I'm doing that is because I'm going to use that circle to extract individual values from my diffraction pattern, and NumPy wants to index them by integer values. So as before, I initialize what I'm going to fill in my for loop, but in this case, instead of getting 2D information, we are going to get one-dimensional information. So I'm creating this line profile, it's empty. And then as I run through all the different dots in my circle, I can extract my diffraction pattern information. This line that's commented out would be the most direct way to do so. You're just going to take your diffraction pattern and take the intensity at that as x and y positions. But I found that in order to get more robust information, it's actually better to do some nearest neighbor averaging, which I'm going to introduce here. So what I'm doing is I'm taking the average, and I'm taking an area now that's going to be 3 by 3 or 9 nearest neighbor pixels. So each one of those little dots in the map below actually represents 9 pixels. You want to create an x-axis, so you have something to plot against in degrees. And then now we're going to plot. You see here, I'm using GridSpec, which is sort of a fancy tool from Matplotlib, to be able to change the height ratios of my plots. And this is something you can look into more as you get more comfortable in Python. And I'm plotting everything below here. So we've already discussed this. We have our ring detector now. So let's apply to the full data set. And all that's going on here in this code is I'm applying it to every pixel in real space. I'm adding one additional thing, which is if you go up here, you'll notice that our diffraction pattern seems to have 4-fold symmetry. And you might not care whether or not your maximum is over here at the orange or over here at the purple. That's more a representation of beam deflection versus really rotation within the sample. So as we adding one extra line here where we are dividing by 1/4 of the number of pixels and plotting the remainder, so then we can get an extra color map. And these maps are going to be shown down here. So this is now we've calculated the location of the maximum for every pixel in real space. But in the middle is this map I like a lot better that accounts for symmetry. And we can compare that to the bright field. We see that within our sample, there's not a lot of rotation, maybe some domains, but little nearest neighbor rotation. And then maybe a few areas where there are outliers, and we can start to ask ourselves, do these areas with rotation, are they lined up where the precipitates are? Is that telling us that the rotation is due to the precipitate and possibly these are areas of high strain? So what we could do with this analysis, if we were to go further and not something for today, is once you start to understand rotation for a sample with a known lattice parameter, you could start to quantitatively assess stress and strain within your system. So this would be a very different technique, but something you built yourself to try and get at some of the information from the cool tools that we saw before that Digital Micrograph has for analyzing strain. So we have one more advanced analysis method. See if there's any more questions, Roberto.
Yeah, just I have one here that for this symmetry analysis, right? Can the code be modified to, for example, plot a kind of a map of the symmetry, sort of, you know, that in this case here have a fourfold symmetry, right? So it's kind of easy to see. But what about if that symmetry changes across the sample? So is that possible to do in a similar fashion?
Totally. And I think that Python is a really good tool to do that, because when you have an idea like that, you can start to think about how to build your own detector or collect that kind of information in a different way. We're about to talk about how you can use a quadrant detector to collect kind of information about what's opposite sides of your sample. But you could imagine a situation where you wanted to build something like a quadrant detector, but maybe with a lot more segments in your diffraction pattern, and you can collect relative intensities in each of those and try and create some sort of map of symmetry within your sample. So that's not something we've explored so far, but you could use some tools like the quadrant detector, or I guess even the ring detector, looking at the thresholding the number of peaks to try and assess that kind of information. So yeah, definitely.
Yeah. And then another question here that I guess you can answer. I can answer that too. If you try to use analyze with MATLAB, the answer is yes. So we use that, use both all the time. Yeah. I can speak for myself. I actually started by using MATLAB, and I just moved. I moved to Python, like say around like 2 years ago. So just basically because one, it's open, like it's free. You don't have to pay for that. And there is more codes available these days in Python, I guess. The whole field is moving more to Python. I don't know what's your, you want to comment on that?
No, yeah, totally the same. I started in MATLAB and moved to Python about a year and a half ago. And there's a lot of already existing code and help online for Python. That's really helpful, but not everything is moved to Python. So I end up doing a little bit of MATLAB and going back and forth between the two.
All right. Yeah, we continue to the last.
We continue. Okay, so the last thing we're going to do is a quadrant detector. And as before, I'll scroll down to the bottom of the kernel to show where we're going and then explain how we got here. So for DPC, you might be familiar with a setup like this. We want to do a subtract quadrant 1 from quadrant 3 and quadrant 2 from quadrant 4. And Python is a really good platform to be able to do this because you have so much tunability over your detectors. So in a traditional DPC setup, you would have just the X and the Y and straight up and down. But what I've done is I've built this specifically so you can tune your outer diameter of your detector if you wanted to bring the radius of your sphere in or out. And then you can also rotate it. So you have two lines here, and they're made, basically the way I did it is by creating lines with point-point form. So I defined a second line, a second point. We know the first point already because we found the center of our diffraction pattern, and immediately that defines our first line. And then using a matrix, I can rotate that by 90 degrees. And then I have two lines that are perpendicular and can use that to divide up my circle into 4 detectors. And then, like I said before, this is very tunable. You could envision a situation where you have 6 detectors to account for 6-fold symmetry, or 24 detectors, or different detectors of different radii. And then also the reason I like that you can change its orientation is here you can see I've aligned it in such a way that none of the lines are slicing right through a reflection. If it was straight up and down, you might have something awkward where a third of one disk was spilling over into the one detector and 2/3 was in the other detector, which is not what you'd want. So let's go back up and look at the code to see how we got here. So this is where I build my rotation matrix to rotate my second point by 90 degrees so I can generate these lines. The reason I left this open is in case you did want to change it from 90 degrees to 60 degrees, for example. And this is my point I've defined here. So XA, YA, and I apply my matrix to get two more values, XB, YB. And then this is where I set the outside of my detector. Even though this looks more complicated, it's no different than what we've already done. We define the indices, you create your circle, and then using lines that are parallel and perpendicular and changing whether it's greater than or less than and iterating over these, we can create 4 detectors. The extra thing I've added here is for each detector, I wanted to find its center of mass. And the reason I'm doing that is so in the bottom image where I plotted, I could label them correctly, 1, 2, 3, and 4. And this type of technique, we're really interested in 1 versus 3 and 2 versus 4, and less interested in 1 and 2 and 3 and 4, comparing those. So it's important to make sure that we actually know which detector is which. As before, I created my own color maps to make the visualization of the masks easier. And then I'm just plotting the 4 masks and adding text at the center of mass of each mask and plotting that over the diffraction pattern. So that's how we got the image below here. The last thing I did as a sanity check was actually to print out the number of elements in each of my masks. You can see here they are all equal, just because I've plenty of time made mistakes and had challenges in making sure that these were the right size or had funny things going wrong. So this is just one last check. So even though our detectors are pizza shaped instead of round, we're going to do the same thing as before. We are going to initialize our image, and then we are going to iterate through all of diffraction space, and applying the masks as binary filters, we are going to extract images from each of those 4 quadrants. So that's what we've done here. This is actually pretty fast in terms of speed of computation. And then what we're going to do is plot 1 versus 3 and 2 versus 4 and normalize. What I'd like to do is to make it clearer what's going on is have 1 versus 3 and 2 versus 4 plotted on the same axes. So the color scale in each of those are the same. So could be fancier or better ways to do it, but I'm doing this manually. I'm just printing out using NumPy to find the maximum and the minimum for each of these 2 maps I'd like to plot. These values are down here. So I'm taking the global max now, which is 0.41, and the global min, which is 0.40, and manually adding them in here when I plot my resulting information, because then I can add it to the same for the second one, and they're plotted on the same color scale now. The last thing I'm doing here is I'm adding insets to my image. So I'm adding smaller axes where I can show the masks, because in this case, we picked our mask orientation and diameter very specifically. So we want to be able to show that. So now we've plotted down here, we've plotted our quadrant detectors 1 and 3 and our quadrant detectors 2 and 4. And we have the masks to show where we're getting the information from. And we can compare them to our bright field image. And this is starting to assess different types of fields within our sample. Based on the information we know about the sample, we assume that this has a lot to do with strain fields. But this is a good starting point for doing some further analysis, combined with the rotational information from before to assess strain in this type of sample. So that's all I had to share. I hope that I've convinced you that coding in Python is fun and easily accessible and very useful for analyzing your specimens. And I guess we'll see there are more questions now.
Yeah, thanks, Stephanie. That was great. That was fantastic. A couple questions here. In terms of like access to the notebook. So I'm gonna post it again on the chat. So I'm just gonna do that right now. You can go to Stephanie's GitHub repository and do that. And then the second question is if this would work directly with the DM, with the digital micrograph. And I guess the answer as is would not. So you have to do a few modifications and then create outside of Jupyter Notebook, like a .py file, basically like copying those the information they have from the kernels that Stephanie showed. So there is a nice and quick tutorial to do that on Gatan's website, and you can use that for like a couple of lines just to talk with the with the DM, and then you can run everything on that. And also the data will be available there, so you can download. If you go to Stephanie's GitHub, it will be there. Right. Is there any other comment or question? So I have a couple minutes. I'm checking the.
Yeah, so just I wanted to add to that. We will, because there were a few people asking, we will share the slides and also links to these. I know we posted some links during the workshop in the chat, but we will also have everything in one place for you, and it will be emailed to the participants afterwards.